Hello students, now we will discuss about simple columnar epithelium. This one, simple columnar epithelium. So cells of this simple columnar epithelium are pillar-like in shape. The cells are tall and slender with actually based means position of a nuclei with basal nuclei. If we draw the structure, then how does it look like? What is function of this simple columnar epithelium? Its major function is absorption and secretion. Function is absorption and secretions. There are different modifications of this simple columnar epithelium. First one, brush bordered epithelium. In case of brush bordered epithelium, if the free end of simple columnar cells are having microvilli, then such types of cells are called as brush bordered, are used with the term brush bordered. If the free end of columnar cells, these are one single layer, so we are using the term like simple columnar, are with microvilli, then called as brush bordered epithelium. If we write the diagrammatic representation of this, how does this look like? So, elongated cells, like this. But as we have written here, the free end must be having microvilli. So like this. Like this. So these are called as brush bordered epithelium. So they must have elongated nuclei. So elongated nuclei. Second one, glandular columnar epithelium. When the free end of, not free end, when these cells are having some secretory glands, example, goblet cells are there, those are mucus secreting type of cells. If they are present inside the cells of columnar epithelium, then we can say such type of columnar cells are glandular in nature. So first we will go with diagram so that it will be easy. This is one pillar-like columnar cell, but the neighboring will be having some secretory goblet cell So it is secreting mucus. So now when this goblet cell is present, then the position of nuclei, it may change because of existence of other gland-like structure. So we can draw the nucleus here. So now here, this is goblet cells. This is mucus or any other secretion. This is columnar cell. So here we did not mention about the location of this brush bordered epithelium. Here, in which types of structures can we see this one means gallbladder. In gallbladder, we can see brush bordered epithelial columnar cells. Now, in which type? What is glandular? If the columnar cells have unicellular mucus secreting goblet cells. Then such types of cells are means location is between the columnar cells or side by side columnar cells we can see within layer. So what is location of this glandular epithelium? In which types of structures or body organs can we see this one? We can see in case of stomach. Next, colon. Next, rectum. And also in uterus. Third one, glandular brush bordered columnar epithelium. Glandular brush bordered columnar epithelium. Here, both microvilli will be present on the free end, and also the goblet cells are also present in some of the cells of one single layered columnar epithelium. Coming to the diagrammatic representation, this will be the microvilli and the neighboring cell will be having the mucus secreting goblet cells, and the subsequent next cell may have again microvilli. Next, again microvilli. Again other cell may be having the mucus secreting goblet cells. So here we are seeing both microvilli and also the secretory goblet cell. Where do we see such type of glandular brush bordered epithelial cells? So example is duodenum and ileum. We'll write just meaning of this glandular brush bordered epithelium. Cells or the layer we'll write. This layer has microvilli on free end and also secretory goblet cells. Next fourth type of cell is ciliated epithelium. Ciliated columnar epithelium. In case of fallopian tubules and in case of some of the cells of brain ventricles, ependymal cells that we call, in such types of areas, the ciliated columnar epithelial cells are present. Okay. Okay, ciliated epithelial cells. These columnar epithelium has cilia on free end. So if we go for diagrammatic representation, these are elongated nuclei at base, and cilia will be present at the free end. So these can be labeled as cilia. This is basal nuclei. So what are examples according to our NCRT? According to NCRT, one example is fallopian tubules and bronchioles also. Inner surface of hollow organs like bronchioles and fallopian tubules. Even in brain ventricles also these are present. We have 4 brain ventricles. So inner lining of brain ventricles and inner lining of bronchioles and also fallopian tubules. Next, we can mention this one starting from inside. So other means one more structure is there regarding the simple columnar cells, that is called as stereociliated columnar epithelium, that is present in the inner lining of vasa deferens and epididymis, of which is the structure which we see in case of testis. So which structure shall we erase? Here we will write the 4th type, 5th type. Stereociliated columnar epithelium. So where it is located? It is located in epididymis and vas deferens. So this type of stereociliated, it is present on the stereocilia. What is use of this stereocilia? The stereociliated means cilia-like structures. These are basically made up of actin protein, and these are majorly involved in balancing of the cells. We'll write one more point. Stereocilia made of actin protein. Actin protein used in increasing surface area. Now, here microvilli also, it supports in increasing of surface area. Next, yes, majorly microvilli structures are used. Why that shape is maintained is to increase the surface area for absorption, because columnar epithelium we have written here, one of the function is absorption. So these are the points that are required for your syllabus purpose.